Well, hello. Welcome to Chapter 20, Part 2 of the discussion about DNA tools and biotechnology. So Part 2, we're going to kind of focus on expressing cloned genes. So remember Part 1, we talked about cloning genes and like cloning vectors where you just want to kind of store and replicate the cloned gene that's inside a vector, or the recombinant vector. Now let's talk about expressing cloned genes, or expression vectors. So what we mean by expressing genes is basically producing the gene's protein product. So this is the process called translation in a cell, or transcription and translation in a cell. So transcription produces the messenger RNA. Remember, messenger RNA holds the code in order to make proteins. And translation is the process of protein synthesis. So expressing genes involves both of those steps. And now it's important to remember that in bacteria and archaea, they're a lot simpler than eukaryotic cells. Eukaryotic cells generally involve another step in between transcription and translation, and that's RNA processing or RNA editing. So RNA processing and editing involves, remember, in eukaryotes, the transcription occurs in the nucleus. So the transcript, or the messenger RNA, needs to translocate from the nucleus to the cytoplasm where the ribosomes are in order to produce the protein. Now, the initial transcript needs to be modified, and this modification involves the addition of a 5' phosphate and a 3' poly A tail, and also, in some cases, the removal of introns. And basically, that's called Rna splicing. So the introns alternatively can be removed, and then the exons are spliced together. So basically, expressing cloned genes, it involves the induction of the start of this process. So what you want to do when cloning genes in eukaryotic genes is once the gene has been cloned, again, what we mean by cloning the gene is that you copied the gene and then you have put it in a vector. The protein product can be produced in larger amounts. So basically, you can force genes or eukaryotic cells or any cell to produce the protein in high amounts. And of course, this can be used for research to study the protein, the structure of the protein, and the protein's function, and for practical applications such as use the protein for medical purposes, like overproduce insulin. Then you need to purify the insulin, and then you could use that for individuals who have diabetes. So cloned genes can be expressed in bacterial or eukaryotic cells, as I already mentioned. And of course, the vector is not going to be a cloning vector. It's called an expression vector. And expression vectors are designed in a different way, and they're designed to induce the expression of the gene that has been inserted into it. So let's look at this one example of an expression system. And this is a bacterial expression system. So it's important to note that here we have an expression vector, and the polylinker is the region where the insert is going to be placed. And here we have a gene for antibiotic resistance. So what must be done, and this is just a review, this is the same thing that actually happens with the cloning vectors. So you want to cut the vector with endonucleases or restriction endonucleases or restriction enzymes. And there are going to be specific ones in which you're going to cut your gene of interest as well. And then the desired idea is in a expression vector, you want to insert your gene of interest in a certain direction, and the direction needs to be in the direction in which expression is going to occur. Remember, transcription must occur from this piece of DNA. So once you insert your desired piece of DNA, now we have a recombinant expression vector. this recombinant expression vector will be inserted into the bacteria. So in order to do that, and we'll talk about ways to do that, but this particular bacteria has been heat shocked. When you heat shocked bacteria and you have expression vector surrounding it, it's going to feel like it needs to survive in some way, and the heat basically kind of creates holes in the membrane, and it allows the bacteria to take up components from the environment. And if you make sure that the recombinant vector is within the environment, the recombinant vector will likely be taken up. So that's the kind of the theory behind that. So once you have confirmed that the vector, the recombinant expression vector, is inside your bacteria, you can grow the bacteria so that it is cultured on a plate and selected for by antibiotic resistance. So you can check this by looking for colonies that are actually surviving on the within the medium that has the antibiotic present. So again, those bacteria that do not have the antibiotic present are not going to survive, and those that do have at least have the antibiotic resistance present, they are going to survive. So then now we have the bacteria which contain the expression vector, which now can be induced to produce the protein that is desired. Now, some expression vectors are inducible, and I've been mentioning this, I've been using the term induced, and some of them are not induced. So those that are not induced, the bacteria is already producing the protein, but some are induced under certain conditions, and this may be desired in the future, so that you apply those conditions and it makes the bacteria produce the protein when you want it to be produced. Once the protein is made and it allows to grow and produce that protein, then you want to extract the protein and or the DNA from the plasmid. So this is a general case, and we're talking about expression systems. So here, let's talk about the protein being extracted. So once you open up the DNA or the cells, you have a bunch of proteins, and this could be proteins that are specific to the bacterium, and of course your desired protein being produced. Once you've opened up the cells, now you have all of this stuff together. Now you have to actually purify your protein of interest. So purification is really the separation of your desired protein, in this case, from all of the other proteins and components within the cell. So the process of converting bacteria in its natural form to bacterium that contains a recombinant plasmid is called transformation. So transformation involves the presence of a bacterium, and we have this bacterium right here, which has its normal chromosome and an extra chromosomal plasmid. So what you need to do is basically create holes or damage the membrane so that something can go through the membrane and into the cell. Now this can be done in a number of different ways, but ultimately we have something that is going to result in the bacterium taking up your recombinant plasmid. So once your bacteria has been taken up, the plasmid has been taken up, you want to rescue the cells so that they survive with your your recombinant plasmid located in it. Now, not all the bacteria in your culture are going to take up this plasmid, but you want to confirm that the plasmid has been taken up, and this is usually done by a selection process, which has been discussed previously in the first segment. But you can filter out, technically, the untransformed bacteria by applying, making sure that you have an antibiotic resistance gene in your plasmid, right, your recombinant plasmid, and then you take the rescued culture Okay, and then you're going to, well, to rescue it, you're going to put it on a medium. So you've heated it, you might have heated it, applied a chemical or some sort of force for it to take up the bacterium, and then you're going to cool it or just put it in a fresh culture and spread it out on a plate. And this plate is going to contain the antibiotic, right? So those bacteria that do not have the antibiotic resistant gene will not survive in the presence of the antibiotic because the antibiotic is meant to kill them. So after some time, at least 24 hours, you want to take a look at your plate, and those colonies that are present on the plate are going to or should contain the plasmid, which has the antibiotic resistance gene. Now you can pick them off the plate and grow more if you wanted to, or you could sequence them, or you could extract the DNA, grow them a little bit more and extract the DNA, and then perform PCR and study the gene if you want. Okay, so let's talk about different ways of inserting the recombinant DNA or the plasmids into cells. And this can be done in bacterial cells, as we've already discussed, and eukaryotic cells, and also plant cells. Now, some types of cells are more difficult than others. So some eukaryotic cells, like yeast, they have a cell wall, and plants also have a cell wall. So some forceful methods need to be used in order to get the cell to take up the DNA. But ultimately, the main ways are to create holes in the membrane, and this can be done by an electrical pulse. It could be done by heat and or cold, chemical methods, and ultimately physical damage using beads. And to shake them up with the beads. So ultimately, this kind of shows electroporation, but ultimately what you want to do is create holes in the membrane so that the plasmid, or in this case, gold nanoparticles, are inserted or taken up by the cells. Another way would be just using needles and a microscope. Of course, this is under microscopic conditions. You have a very, very small needle and you can insert DNA. So this is a method of cloning whole organisms in this case. So this is a micro injection needle. So this is an actual needle that's piercing the membrane, and then you're basically inserting the recombinant DNA that way. Another more forceful method is microprojectile bombardment. So this is using high pressure in a gun type format, and this is often used in plants because plants have a very rigid. So you could express genes across species. So you can take one gene and put it in another. And if you review the Chapter 17 review videos that I have posted in this module, you'll see how the genetic code is standard across different organisms. So you can take one gene and put it into another type of species, from one species, and put it into another species, and it will be expressed. So once inside the cell, the DNA is going to be incorporated into the cell's DNA, sometimes by recombination. So, it could be in a plasmid type format, which won't be done by recombination. But let's assume that we have DNA that's going to be incorporated into the cell's DNA, and this could be done by recombination that the cell naturally performs. So an example would be the PAX6. It's a gene that actually directs formation of the eye in vertebrates. So if we take PAX6 and compare it to the same gene in flies it directs the formation of the insect eye. So vertebrates and insects are different groups of species, completely different. So remember vertebrates from our studies on taxonomy. The vertebrates are going to be a very large subgroup of chordates, which are deuterostomes, and insects are not deuterostomes; they're protostomes. So they're in a very different group. However, they are still animals. But we can see the fly eye. And what's interesting is the PAX6 genes in the flies and the vertebrates can substitute for each other. So basically you can put the PAX6 genes from vertebrates into flies or from flies into vertebrates, and they will still perform the same function. They will be expressed, and then the eye will form. So let's study study gene function, and you can study gene function by analyzing its expression. And how do we analyze the expression? So analyzing gene expression can involve several different methods, but we'll only talk about a few of them in this segment. So you can identify the presence of messenger RNA. So remember, messenger RNA or RNA is the product of transcription. So that's really the start after the first step of gene expression. And you can identify these messenger RNAs by a method called nucleic acid hybridization. So messenger RNA is a nucleic acid, and you can use complementary molecules with probes, or they're called probes, but you can have them, they could be labeled with something that would serve as a method of detection. So it could be something that generates light, that could be fluorescent. These complementary molecules can be DNA or RNA. Remember, complementary molecules only need to bind or recognize their complementary sequences by hydrogen bonding. So there's also another method which is very similar. It's called in situ hybridization. So this is basically hybridization within cells that are living, right? So you're not opening up the cell, it's the whole organism that's still living. So you could use fluorescent dyes which are attached to the probes, which could be DNA or RNA, and then they're going to go to the location where they're complementary, and you could use different colored fluorescent dyes attached to different probes at the same time to identify the locations of specific RNAs, which means the location of expression of that gene within any tissue. So if we take a look, we'll see an example in just a moment. So again, like I said, different probes can be labeled with different fluorescent dyes, so you can identify multiple genes and or their products within the organism. So this process involves the following. So you have the probes right here, and you can see that this probe has this particular sequence, and then the one probe on the right has a different sequence. So they're going to recognize different genes. So this one will recognize the WG gene, and this one will recognize the EN gene. So it's going to bind to a specific region on that gene within an organism. And in this case, we're looking at the Drosophila. It's a fly. So this one is labeled with a yellow fluorescent dye, and this one is labeled with a blue fluorescent dye. But you can look at multiple genes so that you have different color fluorescent dyes like orange and red. And then when we look at the product here, we can see where these genes are actually expressed. So here we can see that these genes are expressed in segments, in each segment, and basically this format right here. And then, of course, we can see green and red and purple, right? So these represent different fluorescent dyes detecting different genes, or the expression of different genes. So it's important to remember that each of the cells of this particular fly larva has the same DNA. However, the genes that are expressed in different tissues can be different in different segments of the whole embryo. So let's talk about the studying of a single gene in terms of its expression. So we can perform what's called, it's a modification of PCR. It's called reverse transcriptase polymerase chain reaction, or RT-PCR. There's another format of it, which is called quantitative RT-PCR, or qRT-PCR. This method uses a dye that fluoresces only when bound to the double-stranded PCR product. So the process involves the production of, basically the detection of messenger RNA, but producing DNA from that messenger RNA by complementary base pairing. So it's like PCR, like where you're synthesizing a piece of DNA, but here you're using RNA as a template rather than DNA. So using RNA as a template rather than DNA allows you to identify the expression, basically the product of transcription, or the expression of a gene within particular tissues. So let's look at the process of RT-PCR, or reverse transcriptase polymerase chain reaction, to analyze the expression of single genes. All right, so let's assume that we have the messenger RNAs from a cell or some tissue. So you've extracted or opened up the cells, and now you have the messenger RNAs. And remember, the messenger RNAs represent the first step in the expression of genes. So whatever messenger RNAs are present that were created using a gene or the DNA as a template means that these genes are being expressed in that particular tissue. So what you want to do first in this process is create DNA from the messenger RNA. Messenger RNA is not very stable. So, and you can't really copy the messenger RNA, so you can only make DNA from it, which will remain stable. So this is called cDNA. It's complementary DNA. So the first step is cDNA synthesis. So now you have the messenger RNAs, now you're going to produce the DNA. And it's produced by a similar process called PCR, right? Similar as PCR. So you're going to have DNA polymerase, you're going to have, reverse transcriptase is the enzyme that actually does it. So reverse transcriptase is an enzyme that actually uses messenger RNA as a template to make DNA. And in this case, it's called cDNA. So, you have your primers, you have the nucleotides, and then it produces these cDNAs. And then the DNA is going to be copied. So now we have double-stranded DNA, and then you can run it on a gel electrophoresis or run it on a gel, and then identify quantitatively, in this case, or semi-quantitatively in the gel format, you can identify how much of the gene that is being expressed. And over time, we have different embryonic stages. So this would be stage, 0 or 1, 2, 3, 4. So these are different stages, and you can see the increase in expression of a particular gene within the embryo over. So let's talk about how the complementary DNA is made a little bit more in detail. So now we've got DNA from a cell or many cells, and inside the nucleus, remember, there's DNA, and then the messenger RNA is going to be produced in eukaryotic cells, or the RNA, and then the messenger RNA is going to be produced from that second step, which really isn't mentioned here, but it'll be in the review videos. It's called RNA processing. So once RNA processing has occurred, now we have messenger RNA that can leave the nucleus and go into the cytoplasm. So what you're actually doing here is opening up the cell and freeing up the messenger RNAs that have been produced in those cells. And now you have them in a tube. So you use the enzyme called reverse transcriptase. Reverse transcriptase uses RNA as a template to make DNA. So what's going to happen is the RNA is going to be copied using DNA nucleotides. Okay, so the RNA is a template to make the DNA. Eventually, the messenger RNA is going to be degraded. And in vitro, you normally purposely degrade it with an RNase enzyme. And then you copy it. And then this is the PCR part, right? So this is the RT part, right, first. And then we have the PCR part. And that's going to actually copy it and make copies of the DNA and increase the copies to billions, similar to that of PCR. Or traditional PCR. So now we have billions of copies of cDNA. And this is complementary. So this is the DNA that is complementary to the messenger RNA that's been expressed in those tissues. So we can analyze multiple genes or the expression of multiple genes at the same time and, and compare between tissue types. So here, let's look at this part of this figure right here. We have genes that are expressed in tissue 1 labeled with a red dye, and then we have genes that are expressed in tissue 2. So these are two different tissues right here, and the genes that are expressed in tissue 2 are labeled with a green dye. So this is basically the expression of many messenger RNAs that that are labeled differently. So if we look at them, and we have this microarray analysis right here, so each of these are different wells, and basically this is going to, each dot is going to be a well containing copies of the DNA fragments that carry these specific genes. So when there's just a red dot, this means that these genes are expressed in tissue 1. And then if it's just green, then the genes that are only expressed in tissue 2. Now, when there's a yellow dot, it means that both genes are expressed. So the overlap of those fluorescent dyes, the output of the fluorescent dye shows up as yellow rather than red or green. So a combination of the two show up as yellow. And then, of course, the black areas represent the fact that neither of these genes are expressed in those tissues. So each of these represent different tissues and the genes that are expressed within those tissues. So the lighter dots, so the lighter green or the lighter red, pink color means that they're expressed at low levels, and the brighter one is going to show up as expression at a high level. So they'll be highly expressed. So if we look at this, we have, again, genes that are expressed in both tissues. This shows that idea right there. And then genes that are expressed in neither of the tissues. So we can use messenger RNA sequencing, or RNA-seq, to analyze the expression of many genes as well. So microarray is a bit messy and laborsome and very expensive. We could also perform this method. So we have messenger RNAs that are isolated from tissue being studied. So we have the messenger RNAs. Once we have the messenger RNAs, the messenger RNA is going to be cut into smaller sizes, or small fragments of messenger RNA. So once the RNA or the messenger RNAs have been cut into fragments, remember, these are messenger RNAs from many different genes, and that's the purpose, is to study many genes. They're going to be reverse transcribed, as mentioned before, using reverse transcriptase into cDNAs. So now we have many fragments of cDNAs that are basically double-stranded copies of the messenger RNAs. And again, remember, the messenger RNA represents those genes that are actually being expressed within the organisms is being studied. So once we have those cDNAs, you can sequence the cDNAs. And now we have fragments of cDNAs, and again, these are going to be randomly cut into fragments, so they're going to overlap at some point. So the DNA sequences will overlap. And now, since we have so many genes and so many cDNAs, we start to create the overlap, and now we can determine the sequence of those messenger RNAs, the full sequence, by using, of course, computers for analysis and combining and overlapping these sequences to determine the actual sequence of it. So there's another method that is very similar to this, discussed in chapter 21, which is our next chapter, which is going to show how you can study like the soil, which is going to have many different organisms, and you can determine the DNA sequences of the organisms that are actually present in that soil sample. So we can study the expression of groups of genes that are actually interacting with one another. So this method, the RNA-seq or RNA sequencing, is a powerful method for many different reasons. But three of the most important or most powerful reasons are: one, it doesn't depend on you knowing the genomic sequence, because you're actually determining the genomic sequence. You can measure the levels of expression over a wide range of samples and genes. And careful analysis will provide so much information about the expression of a particular gene. So the caveat here is that expression of an individual gene still needs to be confirmed by RT-PCR. So let's say we have determined by RNA sequencing that we have the expression of all, like many different genes. It still needs to be determined individually just to confirm by RT-PCR. Now let's talk about in relation to gene function. So how do we determine the function of genes? So one way to determine the function of a gene is to basically disable the gene and observe the consequences of that. This is called knockout or knockdown. So another way is to perform in vitro mutagenesis. So you can mutate the gene and see its put it back into the organism and then see its effect. You can silence genes with RNAi or RNA interference. So this is a form of knocking down or knocking out genes. And then another, more recently form of editing genes, you can edit in vivo or edit them and then make it in vivo. This is called the CRISPR-Cas9 system. So this allows you to knock out or repair a gene, alter the gene, and you can, what it can do is allow you to drive genes or engineer genes to be favored. So you would, in this case, this is in relation to what we've talked about all semester with natural selection. So you can engineer a gene so that it is extremely beneficial, so it's more likely to be selected for. So let's talk about gene therapy using a retroviral vector. So this is something that's been studied. There's been controversial results on this in the past. However, our methods have improved, or scientists' methods have improved. So there's better outcomes with this process. So let's say we have a gene, we have you want to insert the RNA of a normal, of a gene that has a normal allele into a retrovirus or another vector. Now the retrovirus or another viral vector, so remember viruses infect cells. So you want to infect the cells with something that's beneficial to those cells. So that's the purpose of that, in order to insert the DNA that's needed. And basically you're infecting the cell, but in a positive way. So we have the cloned gene. Let's say it's a normal gene, as opposed to one that is a mutant and causing disease. So we put the cloned gene into the viral genome. In this case, this shows viral RNA. So we're basically allowing recombination of the cloned gene into the viral RNA. And this is going to be packaged up into a viral capsid. Of course, the viral capsid allows the virus to be functional and actually infect the cells. So if we inject this recombinant virus into, let's say, bone marrow cells that have been removed from the patient and cultured, so now we have a culture of eukaryotic cells, and we let the virus infect those cells. When the virus infects those cells, the viral RNA is going to be converted to DNA, and then the DNA is going to be inserted into the chromosomes within the genome of those cells. Once that occurs, now these cells have been basically transformed. This is called transfection. And they've been transformed to normal cells where they're going to be producing normal proteins rather than the mutant proteins, which are actually causing disease. And then we inject the patient in the bone marrow. We're going to inject those normal cells into the patient and see the effects, right? So hopefully the effect will be that, the majority of those cells in the bone marrow are going to be reproduced containing the normal gene so that no disease is caused. So there are other methods for studying gene function. Genome-wide association studies, and this is to test for genetic markers or sequences that vary among individuals. It's these sequences that basically cause individuals to have different, let's say, reactions to pharmaceuticals or different responses to infection. And one way to do that is to detect these SNPs. right? So they're called single nucleotide polymorphisms. These are also sequences that vary among individuals, but however, SNPs are single nucleotides that vary among individuals. Now, if you could do a genome-wide study, say take an individual's whole genome and determine the SNPs and then find the SNPs that they have, the variation of the SNP that they have that actually is suggests that they would likely, let's say, respond or not respond or have a negative response to a particular medication, this is a form of personalized medicine. So if you can take the person's genome, analyze the SNPs, and determine what mutations or variations that they have which are associated with a positive or negative response to a medication, now you can design a treatment for that particular individual based on their specific genetic composition. So, there's a lot of studies right now, and we're working towards this ability to perform personalized medicine. So this kind of shows an example of a SNP. So here we have a SNP that's linked to a normal allele causing no disease, but then let's say this particular region in some individuals, instead of an A and a T base combination there, it's a C and a G. And some SNPs are associated with particular diseases. So if you can detect these SNPs, you can determine first whether an individual is going to be predisposed to have a disease, or this could be maybe not a disease-associated allele. This could be, let's say, a negative response to penicillin, right? So let's say we found the allele that causes a reaction to penicillin in those that are allergic to it. Right? So if you can predict a person's outcome from a treatment, then whether it's going to be positive or negative, you can design the treatment that's specific to that individual. All right, so let's summarize this particular segment here. We talked about expressing genes and the studying of expressed genes, basically identifying them, how gene therapy can be applied, and what SNPs can be used for, and genome-wide studies and personalized medicine. All right, so this is the conclusion of segment number 2. We'll move on to segment number 3.